Hello everyone. So today we're going to focus on the NCE revision for biology subject. It is a simple guide for a better approach to your revision. We're going to start with the chapter Nutrition in Plants, and by the end of this video you should be able to state that photosynthesis is the process by which green plants manufacture their own food. Write down the word equation for photosynthesis. You have to describe how a leaf is adapted for photosynthesis. Examine a cross-section of a root, stem, and a leaf as seen under the microscope. Identify and label xylem and phloem tissues in different transverse sections. And identify and label the different tissues in a leaf. Furthermore, we have associated different concepts such as diffusion and osmosis in this chapter, but we're going to just focus on diffusion. List the factors that are essential for photosynthesis to take place, as well as conduct simple laboratory experiments to show the importance of these factors for photosynthesis. Firstly, what is photosynthesis all about? So plants are known to be autotrophs. Auto means self and troph means the feeding level of an organism in a food chain, for example. So for photosynthesis to happen, we need carbon dioxide, water, light, as well as chlorophyll. So on this screen, you can see that carbon dioxide, water, and light are all needed for photosynthesis to take place. And photosynthesis happens in the chloroplast. So chloroplasts are organelles that have the pigment chlorophyll that traps light energy during photosynthesis in a plant cell. So here you can see a little diagram showing what are the gases needed for photosynthesis and what are the gases involved in photosynthesis, which one is released and which one is taken in. A plant also has a transport system, and it consists of two vessels known as the xylem and the phloem. Both of them construct the vascular bundle. So plants have a support system of vessels that run up and down the plant carrying materials. Just like us human beings have blood vessels, plants also have transporting vessels, and you need to be able to identify where is the xylem and where is the phloem throughout different parts of the plant. Let's start with the xylem. So a xylem vessel is a long hollow tube, meaning that it is empty, stretching from the roots to the leaf. The xylem vessel is a structure made up of many dead cells. So at the beginning, when it was still building, it was a living cell. When they interconnected, and then eventually it lost all of the organelles that it used to have. And the structure is made up of many dead cells as a result. So the inner walls of the xylem are strengthened by the deposition of a substance called lignin. Lignin may be deposited in the form of rings or spirals, as you can see here. So xylem has two main functions. For your exam, you have to state that they conduct water and dissolved minerals from the roots to different parts of the plant, as well as provide a mechanical support for the plant. This is why when the wind's blowing, most of the plants would just be bending back and forth and will not break. That's because of the xylem. Let's move on to the phloem. The phloem is going to conduct or transport manufactured food from photosynthetic cells. So it's going to transport sucrose and amino acids from different green parts of the plant, especially from the leaves, because this is the main structure where photosynthesis will be taking place. So you should be able to recognize the phloem. Just for your general information, general knowledge, you have a cell annexed to it called the companion cell. And this companion cell is going to load all of the manufactured products from photosynthesis into the phloem. So here's a longitudinal section of it, and here's a transverse section of it. How does a plant actually absorb water? So absorption of water takes place through the root hairs. Each root hair is a fine tubular outgrowth of an epidermal cell. It grows between the soil particles, like hugging the soil particles, coming close into contact with water surrounding them. So you have a thin film of liquid surrounding each soil particle, which is also a dilute solution of mineral salts. So the root hairs have a large central vacuole that will be containing the cell sap, and all of the liquid that it is going to absorb is going to be stored there. So the sap in the root hair cell is a relatively concentrated solution of sugars and various salts. Thus, the sap has a lower water potential than the soil. When you're going to measure water, the value of water, we're going to refer to it as a water potential. And the highest value for water potential is 0. So these two solutions are separated by a partially permeable membrane, called the cell membrane, of the root hair cell. Water enters the root hair by osmosis. You need to know what is the process of osmosis, just lightly for this chapter, so just understand that it concerns water and the transport of it through a partially permeable membrane. The entry of water dilutes the sap, and the sap of the root hair cell now has a higher water potential than that of the next cell. However, the water passes by osmosis from the root hair cell to the inner cell and then up. Similarly, water passes from the cell into the next cell of the cortex. This process continues until the water enters the xylem vessels, moving up the plant. Let's take a look at the transverse sections of the root, stem and leaf. You need to recognize the location of the xylem and the phloem. It's quite easy. If you're going to start by the root, as you can see, there's an X structure. X for xylem, and then the surrounding ones would be phloem by default. And now you move on to the next. So since X is in the middle, xylem is expected to be in the middle as well, and then the phloem as a consequence is going to be outside. Then when you're moving again up, you have the leaf, which is the xylem on top, and the phloem at the bottom. But that depends. When you're going to flip the slide in different directions, xylem could be at the bottom and phloem could be at the top. Now let's take a look at what does it look like under a microscope. So we have simplified the diagram in the previous slide like this, right? But then when you're looking at it under the microscope, this is what you really see. And you don't exactly have the X structure. However, you can recognize the xylem because it is very thick, as you can see here. So that's the deposition of lignin, and you're going to recognize it as a xylem. And as a consequence, around it would be the phloem. That's the only two structures that you need to remember. Now let's take a look at the vascular bundles under a microscope. So this is a slide of a stem, and as you can see, it does look a bit like this. What we have done is we have just separated different structures. We have grouped structures that look similar and just drew a line. And if we did the same here, we would have this kind of picture. So here's the xylem, again, because you have the lignin deposition, it's very easy for you to recognize it. And on top, you would have the phloem. All right? Moving on, let's just take a look at the leaf structure. So from the previous grades, you have learned that cells can be animal cells or plant cells, and the different structures of the plant cells. So when you're looking at the leaf closely, you would have different layers of cells, right? The first one would be the epidermis, and then you would have the palisade mesophyll cells. You need to recognize those structures and spell them correctly. Then you would have the spongy mesophyll cells. In between here, you would have an airspace that is filled with moisture. And here you would have the vein where you would have the vascular bundle that is consisting of xylem and phloem. You have something called a stomata, and the stomata is made up of two word cells, as you can see here. So carbon dioxide is going to diffuse through those pores, and oxygen is going to diffuse out as a byproduct. So if we take a close look at the stomata, on an everyday basis, a plant is going to make a decision. Am I going to open my word cells, my stomata, to allow oxygen to get out after photosynthesis and carbon dioxide to come in for further photosynthesis? But that depends on how much water do I already have, because water vapor tends to leave or come inside, depending on how much moisture of air do we have surrounding the plant. So usually the plant will be losing a lot of water vapor when it's going to open the gourd cells, right? So we need to ensure that we have enough water to allow this to happen. And this is going to happen through the process of diffusion. Diffusion is the movement of particles or molecules from a region of higher concentration to a region of lower concentration, usually down a concentration gradient. Now let's take a look at the leaf under the microscope, and again, if you have been able to spot the lignin, you probably easily recognize that this is the xylem, and just beneath you will have the phloem. And here's the stomata. Now let's take a look at how the leaf is adapted for the process of photosynthesis. First of all, you need to be able to recognize all of the structures of a leaf. And the leaf is the organ specialized just for photosynthesis, so there are many structures that allow a leaf to carry out this process. First of all, because of the stomata, we have already visited why. The xylem, the phloem, the gourd cells, as well as the chloroplast, which contains the chlorophyll pigment. Transpiration is not exactly in your syllabus. However, you need to understand how does a plant lose water. So green plants are continuously absorbing water from the soil through the process of osmosis, right? Not all water absorbed will be used by the plant. So the excess water together with the water produced by the cells during the tissue respiration, remember respiration is not breathing in or breathing out. Respiration is about breaking down food to release energy. So the water excess is mainly removed when it is evaporated from those parts of the plant that are above the ground. The loss of water vapor from aerial parts of the plant, especially through the stomata of the leaves, is known as transpiration. And transpiration pull is the suction force caused by transpiration. It is the main factor that causes the water movement up the xylem. And as you can see here, it just shows you how the roots absorb the water by the process of osmosis. And then the water molecules will be sticking to each other and to the walls because of cohesion and adhesion. So the water molecules will be adhering to the walls and cohere to each other, and eventually get evaporated from the stomata, which is going to open because of what? Because it needs to exchange gases. So it needs to release oxygen and bring in carbon dioxide. So what are the factors affecting the rate of transpiration? Rises in temperature increase the rate of evaporation. Hence, transpiration also increases strong winds, reduce the moisture surrounding the leaves. High humidity, which is water vapor in the air, decreases the rate of transpiration. Light affects the size of stomata opening, and hence will increase the rate of transpiration. So you have all of the factors listed here, and you can see how water is getting evaporated, like moving out of the leaf. Now, let's try to investigate on a few things. Let's get to the lab. Light density on the rate of photosynthesis. Photosynthesis is a process that is very difficult to monitor in a garden, for example. Hence, we use the evolution of oxygen bubbles to show how fast or slow the photosynthesis process is happening. Here we have a setup experiment where you have a beaker filled with water mixed with a solution of sodium hydrogen carbonate to ensure a good source of carbon dioxide, an aquatic plant under a funnel and a boiling tube. We're going to vary the distance between the beaker and the light source. For this, every single time we're going to move the light closer, we will need to record the distance between the light and the beaker. For example, now it is at 1 meter away from the beaker. We could record that in a table and then eventually build a graph from the data obtained. We shall eventually reduce the distance by 10 centimeters and record the number of bubbles released for 1 minute. For this, you will need a stopwatch. Next, we're going to investigate the presence of starch in photosynthetic leaf. So, the presence of starch in a leaf indicates that the manufactured food from the process of photosynthesis has been stored as starch for later use. starch is more compact and more convenient to be stored than the food being manufactured during photosynthesis right away. Why? Because it's not going to interfere with the osmotic potential of the cell, and it's more compact to be stored. So the procedure involves that you place the leaf in a boiling water. To remove the chlorophyll, you're going to place that in ethanol. Be careful because this is a flammable substance, so usually we should be moving the beaker away from the Bunsen burner, and we can just place the boiling tube with the ethanol and the leaf away from the flame. You remove the leaf from the ethanol and then you place that on a tile. You spread the leaf. Eventually you just add a couple of drops of iodine solution to test for starch. You shall expect the iodine solution, which is yellowish brown in color, to change to blue-black in the presence of starch. The next investigation consists of the movement of water through the xylem. There has been questions in the past where you place a plant material in a dyed solution and then you try to spot which section of the plant has been, transporting the dyed water. You would expect the dyed section of the plant material to be mainly xylem, right? Because it transports water and minerals from the roots to other parts of the plants. So here, as you can see, this is a cross-section view of the celery under a microscope, and when you magnify it, you can easily distinguish where is the xylem from the rest of the other tissues. And this is how you would have drawn it if it was a biological drawing. Investigating the importance of light for photosynthesis is a very interesting experiment. You would have two plants. So plant A is considered as a control in this experiment as it has all the conditions needed for photosynthesis to happen, and it is used for comparison with the other plants. First of all, we're going to take both plants and place them in complete darkness under a black hood, just like plant B, for approximately 48 hours. Why are we doing this? Is just to remove any starch that has been produced previously by photosynthesis. And then we're going to remove plant A from the darkness and expose it to sunlight for like 2 hours. Here we're using a lamp, but plant B is still under a black hood. So we're keeping this as a control here. And then we're going to pluck a leaf from each and then do this starch test as we have performed in the previous experiment. You would expect starch presence here, where you would see iodine solution would change from yellowish brown to blue-black, and here absence of starch, where when you would be adding a couple of drops of iodine solution, it shall remain yellowish brown in color. Then the importance of carbon dioxide for photosynthesis. So here you have a potted plant that has been discharged for like 48 hours. Leaf A is still attached to the plant and is inserted in a conical flask containing potassium hydroxide. You should expect absence of starch in this kind of leaf because the potassium hydroxide has removed all carbon dioxide in the flask, while leaf B has all the conditions required: sunlight, water vapor, water, and then you have chlorophyll. And carbon dioxide should be whatever carbon dioxide has been inside is sure enough to carry on with photosynthesis. Then you're going to plug both of them and then perform starch tests. As you already know, this one is going to show absence of starch, so it's going to remain yellowish brown when you're going to add iodine solution, and this one is going to turn blue-black upon addition of iodine solution. Next, we're going to look at the importance of chlorophyll for photosynthesis. There is a type of plant called the variegated plant where you have different distribution of chlorophyll on the leaf. So here, if you take this leaf and you perform starch tests where you're going to repeat the experiments that we have done in the previous slides, and then you're just going to put that in boiling water, then eventually in a boiling water bath with ethanol, and then add the solution of iodine, a couple of drops of them, you would see that wherever there was the green part around the edges, this is the only section that is going to turn blue-black. So this is showing presence of starch. A variegated leaf is one which has patches of green where chlorophyll is present, and white or other colors where chlorophyll is absent. We have come to the end of this lesson. If you have any question pertaining to the concepts addressed, please feel free to comment below and I shall get back to you. Thank you for being kind and respectful in the comment sections. All the best for your revision.